Good afternoon, also from my side. I'm really glad that Regan and my previous speakers have already paved some information, so we don't have to start from scratch. My topic is the natural history studies. I think we've mentioned these natural history studies and the importance of it many times in the last couple of hours. And I want to go through what is a natural history study and why do we need these natural history studies? What do we already know? And what information is needed to further help pave the way to trials? How do we get to the information we need? And where are these studies conducted so that you know what is happening and what are our ideas to get this further. So these are the questions I asked myself when I prepared this talk. So as you've heard before, LAMA2 related muscular dystrophy is a spectrum of a disease, and it goes from the severe form of congenital muscular dystrophy with a complete myosin deficiency to a much milder form with a more limb girdle presentation, and there is also intrafamilial variability that we don't completely understand why this happens. Laminin alpha 2 is expressed in many different tissues, and this explains why we don't only have a muscle problem but also other things. And what has also been mentioned before, that we don't actually know how frequent LAMA2 is, and I think this knowledge will help us to get further if we get more information on that. Not only how frequent the disease is, but also how frequent is the severe form, how frequent is the less severe form, because that helps us to plan trials or to find the ideal population to actually test all these compounds that were just presented in the last talk. So when you look at these different numbers, for Switzerland, that would mean we would have between 11 or 200 patients in Switzerland. This is a big spectrum, and we actually found 17. So we certainly missed some, but I think this is one problem that we have at the moment, that we don't get access to all the patients. And what is also important about this prevalence data is that depending on where you live and how the health system is organized, the doctor that you see may not have seen many LAMA2 patients. So you always have to keep that in mind. And this is also something that we can, if we are all together, or if you are all together, I think then we can bring a lot of things to forward for improving standards of care. So why do we need natural history studies? To actually better understand the disease. We now know quite well how the severe form evolves and how it presents, but we don't know that there are some in between. They are not all the same. So I think we need this information, because when we start a trial, we need to know how we think that this patient would evolve without the medication. If we want to improve clinical care, we want to know about prognosis. We want to know if we see a child, how is this child, individual child, going to evolve? And we can only now say, okay, this is maybe in this direction, but we cannot give exact prognosis. We need to know about possible complications. I think we know quite a lot on the severe forms now. Regan has already presented quite a lot on respiratory and cardiac and so on. So I think it is important to know about these complications, to be aware of it, to maybe treat early or look for them early enough, but just to be one step ahead of the actual disease to help that. And if we know more about the disease and more about the spectrum, it's also easier to diagnose a patient who has maybe not the completely typical phenotype. So this is also something that we will learn when we have more patients or more information on more patients. And at the end, this can also give information to have more detailed standards of care. Regan has already shown the standards of care of congenital muscular dystrophies, but it would be nice to have LAMA2 standards of care. Like we have in Duchenne, like we have in SMA, and I think this is also, I'm going to go back to that. We've learned quite a lot from SMA and Duchenne, and I think this is a good thing because we have learned a lot, so this should be easier now with LAMA2. And we can have harmonized standards of care. And if we have harmonized standards of care, every child is treated similarly. And if we do a trial, they all have the same standards of care, and we have a uniform treatment before that. This has already been shown, so that was a publication, and that has been like translated into a really nice family guide, which can be accessed via websites, also in different languages, which is really, really helpful. Also, sometimes to discuss with your treating doctor if he doesn't have much, or he or she doesn't have much experience with LAMA2. Now, why do we need national natural history studies? I think it is important to combine all the national studies, but it's also important to have national registries or national studies that we get information on the epidemiology. Maybe there is regional differences, that some founder mutations, that some special phenotypes are more prevalent in one region than in another. This also helps if you have a national network. This also helps to have at least a harmonized standards of care within the country. And also that you know each other, the physicians working together know each other. So there's an exchange, and this again helps a lot. And it gives the possibility to have national questions answered as well. Now, what do we know about myosin deficiency? I think I go quickly through it. It's some repetition from what Regan said. So we know it's usually a weakness or hypotonia at birth or within the first months of life, markedly with delayed motor milestones. Many of the complete myosin deficient patients never reach the ability to walk, and the contractures develop quite quickly. Usually that starts at the elbows, hips, knees, long finger flexors, and sometimes that is even more important than the weakness. We have marked axial weakness, especially the neck flexors and the upper arms, and a rigid spine evolves, and a hyperlordosis, which on one hand, as Regan said, helps to stabilize while sitting, but on the other hand also leads to a position in sitting which is not really good for the evolution of this hyperlordosis. So the patients lean forward more and more to have this stability, but the lordosis gets worse. There's a facial weakness, often a macroglossia and malocclusion problems. We've seen this picture, and this is just all the patients are really, they are similar. They have this very optimistic way to look at life, and they have I don't know how to. Can you make the middle video? Yeah, thank you. And I have this interesting way to shuffle, which I've only seen in LAMA2 patients to be done like that. Probably also because of this hyperlordosis and so. And if the hyperlordosis gets worse and the contractures get worse, children usually have this position in the wheelchair, which is seems to be very comfortable, like the girl there, but it's also not very good on on the progression of the contractures, because it's actually right in the contractures, and they don't like if we want to sit them correctly. They want to get into the position that is comfortable for them, and this is quite understandable. We often have a failure to thrive, and also even if they have a gastrostomy. Ophthalmoplegia is a frequent thing. All the patients develop progressive restrictive respiratory dysfunction, and cardiac involvement has been described. And back to the brain involvement, seizures are, it's interesting that the brain MRI changes are very often, but the seizures are not that often. So you have 10 to 30% of the patients develop seizures, some only during a certain time and it evolves. Some have very easy to treat seizures, but then there are some patients who are really difficult to treat seizures, and we don't completely understand that either. So this is also something that we would need to know about the natural history. Usually the cognitive function is normal, but then again, there are patients with autism, there are patients with a cognitive problem, and also that is still not really well defined, and there's also work needed. And this is just to illustrate these brain changes, and I just want to show you some pictures. This is two slices through the brain, and what you see here is this so-called white matter, and usually this is dark, this is gray, and this you can see that this is much too light, and also here. And what we see also often is that the cerebellum is just a little bit too small. And what we can see also quite often is that not only the white matter is changed, but you can see here, normally the cortical formation is like that, but here you can see that it's just like a band and a bit thicker, and this is probably, what we see in the MRI where the seizures come from, and probably the malformation in the brain, this is what we see, but probably if we would look more closely, then we would find more, which probably explains why not all of the patients who have seizures have these malformations. And on the other hand, many of these patients who have the malformations don't have the seizures, and we don't understand exactly why. This is just that these white matter changes are more prominent, are often not seen at birth. So if the MRI has only been done at birth, this does not mean that it doesn't have any white matter changes that evolves. This is at 2 months, we can already see it. And again, here, there's also more changes. Sometimes there's even a hydrocephalus which needs a shunt. This is quite rare, but also, and this child actually has a polymicrogyria, has this ventricular megaly, needs a shunt, has a small pons and a Blake's pouch as the reason for the hydrocephalus, and his cognitive development is absolutely normal. No seizures. And this is what I wanted to show about the scoliosis. What we know is we have a rigid spine. We have the risk to develop a scoliosis, and usually also this very stiff hyperlordosis. And I think this is something that impedes the motor function as well. So this was at the age of 9 years. She was already quite hyperlordotic, but no scoliosis. And then there was a growth spurt, and she got this scoliosis, and this hyperlordosis got worse and worse, so that she was not able to sit independently anymore because she was too much hyperlordotic. And now she got, she had to have surgery, which went well. and we'll see has already a better sitting position now. But this is always a very difficult decision to operate, and how far. So this is also something that we have to learn, which is actually the best way to deal with the scoliosis and the spine deformities. The spectrum, as we've heard before, of this disease is quite wide. We have then milder phenotypes, and we have very mild phenotypes. I know a now adult woman who's probably online, then I followed for quite a while, who has no weakness, just a bit of calf hypertrophy, some TI tightness, some muscle pain while exercising, but otherwise no weakness, no contractures, and she has the absolute typical white matter changes in the MRI, but she is very bright and is now at university. So this is really a wide spectrum, and we need, before we want to try any medication, we need to know what we deal with. Now, what do we need? What do we know about natural histories? This has been shown yesterday. That was a retrospective study done in London, where you see the elbow. This was mostly severe forms, where you see that the elbow contractures get worse. Again, the knee flexion contractures get worse, not as clearly as this. And the scoliosis, how that evolves, usually after the age of 5 years until puberty, you can see that the Cobb angle gets worse. And this is without scoliosis. So this is actually something that gets less and less. And they've also looked at motor functions like sitting, walking, and looked at it just at one time, and also for each patient individually, how they evolved. And also this natural history study that was done at the NIH, Regan has already mentioned it. And you see here, this is the LAMA2 patients. And here you see when you do a certain test that the test gets worse. But the problem here is, so this showed that one of these scores is really showing a decrease. So this is something that we can use for future study. But with only this, we cannot test in a clinical trial. We need much more data in a much bigger range of age. So why do we need this natural history study? We want to be ready for trials. And if there is a medication and we want to prove that it helps, we need to know what are the inclusion and exclusion criteria. In a trial, we want to have a population that is relatively similar, where we know how they would evolve if we don't do anything. And then we say, okay, this population usually behaves like that. And now we have a medication and it behaves like this, and then it's much clearer to show that that it actually works. But this is ideal world, so I think we do need all this information to actually help develop clinical trials. We need to have measurements to evaluate the effect, and we need to have the information on how often do we have to test these patients to see an effect. It doesn't make sense to have a test every 2 months if it only shows up after a year. This is just too much work for everybody involved. We also need the natural history studies, because then we know where the patients are and we can quickly find them if they fulfill a certain inclusion criteria. And this is also something that helps feasibility. If a big pharmaceutical company wants to do a trial, they want to know how many patients can you include in the trial quickly. Because the longer it takes, the more expensive it is. So that if we are ready with that, this is also something that helps. I think what we learned from other diseases is that in SMA and Duchenne, the registries where this information was stored has greatly helped to recruit for trials in SMA and Duchenne. I think this has helped a lot. And actually, what we also learned is that when the first medications came into the field, we were not so good in outcome measures. We didn't have the right outcome measures. We didn't have enough natural history study. So now that all these compounds are in the pipeline, if we are quickly doing all this natural history work, I think then we can help to design better trials that actually show quickly if a drug has an effect or not. And of course, the better and clearer we can show an effect of a compound, the faster the approval comes. And we've learned from the SMA field, if a medication is really showing a clear effect, it goes quickly. It goes really quickly. And this is something that is, I think, reassuring. So it goes quite a while until when these results are not clear, but if it's clear, then I think it goes relatively fast. And this is just an example that it can also help to have natural history data. This is SMA. So if you have good clinical natural history study that can even be used as a comparator in clinical trials. And that was the Firefish trial. That was the third compound being developed for SMA. And they actually published the paper and had from the natural history studies that have been done before, they had like this information and didn't have a placebo group. So how do we get to the information we need? There's two possibilities or the mixture of both. We can have registries or we can have dedicated natural history trials, and they have both their pros and cons. In registries, we can only collect regular data that we collect during regular consultations. But we can collect this data as long as we want. It's just there and we can analyze it. All patients can be registered if they have all examinations or not. Everybody is in it and can be found if there's a trial. There's less funding necessary because the registry is funded by several funding agencies, and then you can include something that has not a lot of extra funding. And you can also compare to other diseases, which is also helpful, or use the same survey for several diseases and so on. And then you have the natural history trials. They are often centralized. This has the advantage that all the children or all the patients are seen in one center with one physiotherapist, and that you have much more exact data, but this is not real world. It can include more investigations, so you can actually see if there's one test better than the other. So you can have a whole list of investigations done, and then after it look what has worked best. This is extra dedicated visits, so this is more work for the patients and the families. And you can do investigations that would not belong to normal consultations. It usually is only over a defined time, which is a negativum, and there needs to be extra funding, because that's really a trial where everything else needs to be funded by the study. And how we do it, we have, this doesn't depend on registry or not, it depends on what. So what we do is when a patient comes, we just, we collect muscle strength, contractures, facial weakness, motor functions, then the whole physiotherapy scores, what were done, and the contractures, and so on, and cardiac investigations and pulmonary examinations and so on. And actually natural history studies are being conducted or planned in a lot of countries already. So I think this is really very reassuring. And what we would like is, or what has been done by Stefano Previtali, is that we, as some people here, we have applied for a new European grant to would allow us to collect data over 3 years in a harmonized data set. And there are other studies running, so we could, the aim of this project is to search also for biomarkers, so that would be an extra thing that we want to see, that if anything in the blood or in MRI or something that can be shown to be correlating with the disease cause, that would help in a trial to have something objective to then see if medication works. Some of the data can be collected as routine data within a registry, but other things, for that we will need to have extra ethics approval and extra funding. So this is really depending on if we get the grant or not. So what are the challenges and where can you help? I think the challenge is to align all these studies, that we tomorrow maybe we agree on the same outcome measures, maybe the same intervals that we look, maybe also to decide, okay, one country does one and the other does something else, and we compare. So I think the network is something that will help. Where you can help is help us find all the patients. I think the more patients that we have in these studies, the more information we get. And what is also a challenge is to find scores across the lifespan and across a large spectrum of severity, as we've shown. We need to examine children who are very weak, and then maybe adults that are not very weak. And if it would be possible to do that all with one scale, we would be very lucky. And then we would have to correlate that with the contractures and the spine deformity. And for the spine deformity, for the lordosis and the neck, there is no validated, so we need to find a common way to grade that. And we need patient-reported outcome measures. So the one thing was the global impression. Has it? Have you the feeling that something is better, worse, or something? But maybe we should need some more meaningful measures. So there is something where the patient and the families can certainly help us getting to really relevant outcome measures. And with this, I want to thank you for your attention, and I'm open for questions.